Good morning. First, let me just thank Allison FAST to support in this project. Also invited me to talk to you about this project. I understand many of you are parents, so I hope I will get a chance to meet you in person whenever it's feasible. So what I'm going to share with you a project supported by FAST, and we just starting about a couple months ago to create a biorepository for Angelman syndrome induced pluripotent stem cell, also called iPSC, and the brain organoid. This is a dream paradigm, I call dream paradigm, to sort of develop a treatment, a novel treatment for Angelman. It can be applied to many other similar to Angelman syndrome. You can see that's many steps you have to taken care of before you get a successful. Let me see if I can use the laser therapy. Oftentimes, you study identify the gene for Angelman. We already identified back 20 years ago, and then you create an animal model. Oftentimes, it's mice and the fish and the monkey and other. Now you're understanding what's the mechanism, why you're missing UB3A gene caused Angelman. And eventually develop a treatment. Could be gene therapy, could be drug and a small molecule, antisense oligo. When you have a new treatment that you need a testing in the animal model, if it's successful, then you can move to the phase 1 clinical trial, then hope eventually you get FDA approved success therapy. So as many of you know, we still understand a lot of detail for Angelman. How do you develop a treatment? This is a cartoon. It's a little complicated molecular nature, it's a detail, but just quickly highlight, this is a gene primarily responsible for Angelman syndrome. If you miss this one gene by many different parts, so the mechanism, but eventually end the deficiency for the UB3A, that's what's caused the Angelman. As many other possibility or mechanism regulate the Angelman, for example, antisense oligo is idea that's antisense nucleotide from long distance and regulate the UB3A, and that's the basis for antisense nucleotide oligo therapy. You can also modulate the protein for the ubiquitin ligase 3A, then develop like small molecule or pharmacological sort of intervention for the Angelman. Here's the kind of concept for antisense oligo, ASL treatment. That's some of your parents are participating and also familiar the concept. Basically, the antisense oligo is capable to reactivate the gene for maternal chromosome, because in almost all the patients, the missing copy is on the maternal chromosome. So I hope I'm kind of correct. It's reactivated the UB3 gene from paternal chromosome because the maternal copy it's either mutated or deleted, or in some rare case, it's a uniparental disomy. So historically, we use a lot of animal model. As some of you may know, when I was a student, I created first the Angelman mouse model, and now has been used for many investigators to develop a treatment for Angelman. So clearly, Angelman mouse model or other animal model are valuable. However, mouse is a mouse. Mouse is not a human. One example shows here, contrast here. This is the human brain. This is the mouse brain. The size-wise is different. And the major difference you can see in the human brain has a lot of fold, and the mouse brain, it's smooth. So that's a significant difference too. So that a lot of challenge facing in the developer treatment is oftentimes the successful treatment for mice and not necessarily translated to the human, because the difference for the human brain and the mouse brain. That's basically the rationale behind this project. Then ideally you want to study human brain or human neuron, which oftentimes it's not feasible in the laboratory. So what's the sort of alternative, and is any possibility we can create a human Angelman neuron or human kind of miniature brain mimicking the human brain? Now you can really testing the idea in the human derived from the neuron. So the first step, oftentimes right now, about 10 years ago, scientists develop a new technique that allow us to do that. So the step one is to harvest the blood cell from AS individual. Then we can use a very special technique, reprogram the blood cell into the induced pluripotent stem cell called iPSC. With the iPSC, we can also use the other method developed later on to let the cell differentiate into the human neuron. And just very recently, scientists also figured out to create somewhere called miniature human brain in a petri dish we call a brain organoid. So what are the induced pluripotent stem cell, iPSC? So basically, it's a special type of the cell, the stem cell, is generated directly from a somatic cell, such as skin cell or blood cell, by reprogramming process. And we call the reprogram because you can introduce only a few genes to the skin cell and the blood cell. This cell will become a stem cell. They can differentiate into, convert it into many different type of cell. It's embryonic type of cell. If you look under the microscope, you can see the cells look like this, which is, this is one single stem cell, iPSC cell. This technique was developed not too long ago, and about 10, 15 years ago, and end up in 2012, where this technique was awarded a Nobel Prize in the medicine, because people see this is a brighter future for this particular technique. Then use this technique a couple of years later, scientists was able to convert some of this iPSC cell to the human neuron. Shows here, this individual human neuron. And what is the brain organelle? Brain organelle, basically, in the short term, an easy understandable term, kind of miniature organ, kind of resembling the human brain, that's created oftentimes by the iPSC cell in the three-dimensional sort of bioreactor. That's kind of new laboratory apparatus. Then over several months of time. So basically that iPS cell growing into gradually became a small size of the brain, which is a diagram here, which you can see. This is a cartoon how this is gonna experiment works. So you study with iPSC cell, and then you through multiple steps. This is a bioreactor. So then kind of culture this cell became going into the brain, the three-dimensional structure, which shows you here. And so that each step eventually, after the mature, you can, by beautiful staining, so you can somewhat see mimicking the human brain structure, is different cell type or different layer, of course, in a very tiny size. And so using this iPSC and the brain organelle, actually my colleague in Singapore and my lab, and published about early this year, we actually shows you on the potential of using or creation this iPSC cell and the brain organelle can be used to understand the Angelman and also testing the new treatment. So for example, this is a iPSC cell we generated from one of the patient. This is the final product for iPSC, and we can also differentiate this one to the new one. It's with beautiful color. So we have the Angelman patient, which is clear missing the UB3A. UB3A is a pink color. We can also bring the UB3A back, so you can see if it is we call rescue the phenotype. We can record in this individual cell and the neuron, and you can see the electric current. And for this cell, which you can see, this is a normal control. This is the Angelman patient. You can see the current. It's more we call hyperexcitable, basically firing much faster, probably mimicking the seizure activity in the human neuron. Then we can also testing the new drug called Paxilin. You can see after Paxilin treatment, then this Angelman neuron became similar to the normal control. Compare here, this Angelman neuron, it's much faster than the wild type normal control neuron. So this is a brain organelle generated from that individual iPSC cell. You can see it's very tiny for the brain organelle from the normal control, and this is a brain organelle from the Angelman patient. We also see a beautiful staining for the different cell type, a different layer of the similar to kind of human cortex. And similar, you can recording from there. This is a control, and then this is the Angelman. You can see firing much faster. Then you can treat it with new drug. Then the Angelman brain organelle basically normalize after treatment with Paxilin. We believe this could be potential treatment for epilepsy and a seizure in the Angelman patient. So clearly, this is the value for the, we show the value for the Angelman iPSC derived the neuron and the brain organelle. However, in that particular study, we only generate one or two iPSC and brain organelle. For really getting this, promoting this direction, we would like to generate a lot more. And so then you can see the, you can do a lot more experiment with replication and for any investigator. So that's Allison and I starting to this discussion about 6 months ago. Then we finally decided that's the good project to do. And we're starting about 3 months ago, right now in December, it will be 3 months ago, that this project at Yale. Again, thanks Fast and Allison for this initiative. So the idea and the plan is to create a biorepository for a panel of Angelman syndrome, iPSC, and brain organelle at Yale. So the objective is to create multiple different cell lines from multiple different etiologies. I know some of the each individual with Angelman could be deletion, could be UB3A, could be UPD, or a few of the patients maybe we call a gain of function mutation. So we will create all different type of iPSC cell first, then later on convert to the new one. Then the plan is also this cell be generated will be able to, we will free to any researcher or academic industrial laboratory to use them. The specific plan is basically, as I mentioned, it's to create multiple different lines, multiple different genotype. And the second is to develop effective mechanism to distribute the cell to any researcher investigator and they're interested in use the cell in the future. So many of you are very familiar with this diagram, and this, as I mentioned earlier, we're going to create a brain organelle and iPSC cell from 5 different, we call the genotype, basically different etiology. The first is a deletion, larger deletion patient from mother's chromosome. Second is a mutation specifically in the UBE3A, oftentimes is disrupted the UBE3A. Then we also collect the UPD patient and also some rare imprinting defect patient. And we're adding another class, which is called a UBE3A gain of function mutation. That's the opposite to the other Angelman patient, which is oftentimes deficiency. We believe this is probably increase the UBE3A activity. It may also not be good if you have too much UBE3A, can cause Angelman-like or primarily sometimes people will say it's more autism-like phenotype. So the project we divide in 2 phases, and the year 1, year 2, we will produce 20 lines, 3 Angelman from 5 different genotypes, and plus 5 control iPSC cell. And the phase 2 will be year 3 or year 4. We're going to produce 8 brain organelle from the iPSC we generated, and from 5 different genotypes, and plus 3 control. So we are just getting started, and thank you. Probably some of you are already given blood and talking to my research coordinator, Cassandra Ding at Yale. I probably met some of you too. I want to take this opportunity, thank you each of you to participate. For any family, it's the first time you heard this project, you want to contact us, please feel free to email me, and also contact FAST, Allison, they will give you the contact information. So specifically, we're looking for individual, right now is age, Angelman patient with age of 5 to 18 years old, both boy and girl will be fine. We roughly want to select some subject with a seizure and subject no seizure, roughly equal. As I said, we will collect 3 AS patients for each class 1 to 5. And we're also gonna create 5 control. We prefer some of the family, if you have a sibling, healthy sibling, you can just donate given about 5 cc blood for this project. To do this project, we are assembling an excellent team at Yale. Myself will be the PI and taking the lead for all aspects of the project. We have excellent team from Yale Stem Cell Center. Some of you know Yale Stem Cell Center created 2006, and it's one of the largest or leading stem cell research center in the world. I hope someday some of family can join us at Yale and to visit the Stem Cell Center. It's a really beautiful facility, and they have done excellent, outstanding work for many, many projects. So the primary and Chai Hong Cho is the IPSC core director. Hai Fan Lin is the director for the stem cell, and Yin Yong Park, he actually is a pioneer for stem cell iPSC cell and the brain organelle in the country. So we're lucky to have a very excellent research team to taking on this project. I'm very confident, and in the 2 years we will be able to create all the cell line that given to all the research and investigator in the world. So again, the final plan is basically on this line when generated. Any researcher in any part of the country or in the world, if they are working in Angelman, we'll be happy to give it to them for free, and as long as they sign agreement, some kind of paperwork with Yale. For non-academic user, basically it's a company, laboratory, and a pharmaceutical company. If they are also working in Angelman, want to access this cell, we just develop an easy agreement with Yale. We will be delivering this to them too. With that, I'm going to acknowledge this people really contributing to the project, and support this project. As many of you, Allison is a wonderful person, and for as also the parents, as you all know, for the Angelman, she and I really are working for quite a long time, and then this is a project to really push for the value for this collaboration. I also thank FAST, the board member, for their support for this project, and the FAST for the support. And most important, I would like to thank many of you, and probably some of you are in the are listening. And so thank you for your participation for this project. I'm looking forward to meet you some days in person. I also would like to thank my colleague at the Yale Stem Cell Center and the School of Medicine for the support. Finally, I'd like to thank the people in my lab, particularly Cassandra. It's the one to leading the coordinate for this project. I think I'm missing one. It's Kwon Young Kim for this slide, and she is the people we're working on this particular project in the lab. Thank you again for your listening, and I hope I'll meet you later today to answer any question you may have. Bye-bye.